Hello, welcome to my presentation on advances in materials and surface characterization of medical devices. This is the talk given at the Biointerface Workshop and Symposium in San Diego on October 3rd, 2017. I will start with a brief introduction to medical devices and then talk of the need for surface and interface characterization in these devices. These relate to performance, safety, and regulatory requirements. Along the way, I will discuss various analytical techniques with examples. What constitutes a medical device? A vast number of products are considered as medical devices, and the level of scrutiny varies on the classification of these devices. These could be simple devices such as band-aids, transdermal patches like nicotine patches, or combination products that incorporate a device and a drug. Materials such as contact lenses, intraocular lenses, and advanced contact lenses such as in-eye glaucoma and glucose sensors, and a whole lot more also constitute medical devices. Certainly, in the near future, we may even have contact lenses that have built-in TV or computer screen, and these are very complex devices. More invasive devices such as needles, catheters, stents, valves, pacemakers with all the batteries and cables that go with it also constitute medical devices. In vitro diagnostic products such as lab on a chip also constitute medical devices since they come in contact with body fluids. Chemistries at the small reaction sites on these semiconductors or glass structures is extremely critical and need extensive surface and interface analysis. FDA has well-defined guidelines meant to have good control over these medical devices with a series of 10993 standards. This slide and a few other slides in this presentation are courtesy of Dr. Anna Ballou from Medtronic. Some standards are well established, some more defined than others. Part 18, which is chemical characterization of materials, plays an important role in material selection. Part 19, dealing with the physical, chemical, morphological, and topographical characterization of materials, is a well-defined extension of Part 18 in the surface properties of these materials. In this presentation, I'll focus more on Part 18 and Part 19 related characterization techniques. Extractables and leachables, surface residuals, and particulates are integral part of Part 18 testing work. Body fluids interact with the devices and extract material that may have toxicological effects. These segregate and exit to and from the surface. Classical surface analytical techniques only give data at a point in time in the material of interest. For example, it is important to know what comes out of a 30-day contact lens over the entire time period. This can be achieved with various combinations of surface analytical and chromatography techniques. It will help in modifying surfaces to protect and inhibit the migration of unwanted chemical entities. For example, titanium oxide coating protects nickel leaching. Now also, residuals from chemical cleaning or processing and particulates on the surface also need to be characterized. There are a host of techniques that are used in extractables and leachables and particulate studies. Some of these are listed here, like gas chromatography mass spec, liquid chromatography mass spec, ion chromatography, and particle size analysis, FTIR, Raman, SEM, TEM for nanoparticles, and various surface analytical techniques. And of course, there is an alphabet soup-filled kitchen sink of techniques that are available. Most medical devices are complex in shape and not necessarily flat. When you use a small area analysis, 250 micron square or 500 micron square, as in most surface analytical techniques, residual solvents and surface contaminants are not easy to characterize because they may be localized in the crevices or areas of the medical device, such as a stent or a catheter, that are not easily accessible to the analytical technique. In this example, the ideal way to characterize the presence of residual solvents after a polymer coating on the stent was with dynamic headspace GC mass spec, indicating the presence of N-methylpyrrolidone. Surface analytical techniques could not detect this since the measurement was extremely localized. Based upon the coating process, there was also a possibility of DMSO, but this was not detected easily with dynamic headspace GC mass spec. With better sensitivity, using selective ion chromatography, DMSO was detected, and these are techniques that are extremely useful when compared to simple surface analytical techniques. Another critical area in device characterization are particulates, whether from the manufacturing process or cleaning process. Classical techniques such as EDS or energy dispersive spectroscopy with an SEM are used for metals, and techniques such as FTIR and Raman spectroscopy are used for polymers. These last two techniques of FTIR and Raman spectroscopy are done in ambient conditions and are very efficient. For nanoscale particles, high-resolution TEM is used. So now jumping to 10993 Part 19. It is a technical document at the present time, but is a surface property extension of Part 18. There are three major aspects to this. They are physicochemical, morphological, and topographical characterization. Important points to remember, as Dr. Anna Bellew has highlighted, Part 19 alone cannot predict biocompatibility or give direct answers about biocompatibility. It can provide a baseline on device surfaces currently under current manufacturing conditions. It can serve as a reference for later on as conditions, processes, and materials change. The next two slides will highlight the techniques that can be used to study the physicochemical, morphological, and topographical properties of these materials. The table lists various properties assessed, such as shape, morphology, topography, surface chemistry, and others. It also lists what it evaluates and some of the most common techniques used. This is an extension of the previous slide. This list is not comprehensive, but is a good starting point to understand the usefulness of Part 19 characterization of medical device materials. There are a wide range of analytical techniques that are used for surface characterization, from morphology to crystal structure to chemical properties. Techniques such as advanced imaging with AFM and TEM and X-ray diffraction for crystal structure or percentage crystallinity in materials are extremely useful and widely used. In the rest of the talk, I'll highlight a few of these techniques with examples and finish off with two new advances in techniques that will play a major part in materials characterization in the coming years. These last two techniques are not presently available at EAG. Atomic force microscopy, more commonly called AFM, is a very useful technique to understand the surface roughness of materials. Classical surface analysis techniques measure the top 5 to 8 nanometers of a surface, and the materials in the crevices and ridges are not easily detected. It is essential to characterize the surface morphology to be sure that we are analyzing what is there. A classical example shown here is of a as received and a used contact lens, and each notch in the Z scale bar is 400 nanometers, indicating that the surface roughness is significantly greater than the analytical depth of common surface analytical techniques of about 5 to 8 nanometers. Imaging and understanding particulate chemistries is also very critical. With advances in technology, contact lenses with colorants and dyes, and as I had mentioned earlier, ones with electrodes and sensors, have abrasive material close to the surface and need to be characterized, and also protected from interaction with body tissue. So, as a worst-case scenario, we used an over-the-counter contact lens that are used for Halloween and have colorants and dyes. A cross-section of this lens shows that there are a large number of particles very close to the surface. We decided to do a bevel cross-section of this surface to expose the area and do time-of-flight SIMS imaging, which is extremely surface sensitive. We can very clearly see the iron pigment and particles close to the surface and the surface siloxane and the HEMA lens material. An RGB image highlights the three regions along with the bright iron particles that are very close to the surface. Nitinol is extensively used in medical devices, and one of the most important requirements is to be sure that there is no nickel leaching. Auger electron spectroscopy has been the technique of choice due to its high spatial resolution and surface sensitivity. This technique along with modern-day electrochemistry, has helped study nickel leaching and inhibit it with surface passivation and coatings. Here is an example of an SEM image and Auger electron image of a nickel-rich region on the surface. Another useful technique is X-ray photoelectron spectroscopy. This technique is useful in giving near-surface elemental and chemical information, and in conjunction with depth profiling, the depth of the surface layer can be characterized. This slide indicates surface scans which show the presence of elements that are detected at the surface. High-resolution scans indicate the presence of a thin layer of chromium and iron oxide on the surface. No nickel is detected very close to the surface. With argon ion depth profiling, the protective oxide on the surface is calculated to be about 40 angstroms deep, and this inhibits nickel from being exposed to the surface. The techniques we have discussed so far have been around for years and successfully used, though there are some limitations. A wide range of surface analytical techniques need ultra-high vacuum. Samples have to be freeze-dried or frozen to protect low vapor pressure materials that may be pumped out in ultra-high vacuum. New technologies in chemical characterization with 10 to 100 nanometer lateral spatial resolution and without the need for ultra-high vacuum will enhance the understanding of medical device surfaces. Advances in these techniques with a combination of AFM and IR and AFM and Raman will significantly enhance the characterization of materials and medical devices in their ambient condition and also in the presence of liquids. The next two slides are courtesy of Kevin Kohler of Anasis Instruments. This is an extremely useful and interesting technique which uses a combination of AFM and infrared spectroscopy. In the AFM-IR technique, the beam from a pulse-tunable IR laser is automatically guided onto a sample surface that is also co-located with an AFM tip. When the infrared laser source is tuned to a wavelength for which there is a corresponding molecular vibration present in the sample, the rapid thermal expansion due to the heating induces an oscillation in the AFM cantilever. The amplitude of the oscillation of the cantilever can be recorded at a specific site while the laser wavelength is scanned over a desired range, producing AFM-IR spectra. This induced amplitude is directly proportional to the absorption coefficient of the material being examined. This enables one to measure IR spectra at extremely small areas down to the 10 to 50 nanometer range. In this example, PEG methyl ether thiol attached to a flat gold substrate through a thiol link is characterized. Topographical images of the selected area is shown in this image, which indicates a non-uniform coating on the surface. Here is the IR spectra averaged over the entire area. The CH2 WAG region in the 1345 inverse centimeter can be extracted at each and every individual pixel, and the regions are then plotted. This gives you a very high spatial resolution image in a very shallow surface depth compared to conventional IR spectroscopy. Tip-enhanced Raman scattering takes AFM and Raman spectroscopy to new levels. This material is courtesy of Eddie Robinson of Horiba Instruments. Co-localized AFM and Raman spectroscopy at very high resolution is shown in this work of a lipid sample. The sensitivity is very high along with very high spatial resolution. To highlight the very high lateral resolution in the nanometer range, this is an example of Raman characterization of carbon nanotubes and graphene oxide using a 638 nanometer laser. Resolutions down to 15 nanometers have been achieved. The beauty of these two techniques is that they are done in ambient condition and can also be done under liquid conditions. In summary, understanding materials and surface properties are extremely essential in medical devices for performance, safety, and regulatory requirements. There are numerous techniques that are available to solve these analytical challenges. The aim is to find the best technique that answers the question of interest or generates new questions. Surface analytical techniques only give information at a point in time on the surface. A combination of analytical techniques is extremely essential in understanding the surfaces of interest over an extended period of time.